let's see. Let me get back on my screen. Okay. So I think everybody who's here is going to be here tonight. Looks like we got a good crowd. We got a total of 15 people. That may be the max, which is good news on a Monday night. All right. So some of you guys, looks like I recognize some of your names. I know Brenda. I think you're one of my students. I've seen you. We've chatted a few times through email. Guys, I kept this chat, and you can either some students like to talk over the microphone, and some guys people like to chat. So if you want to, I want to ask you guys if this is your first or second spotlight session. Chat me something. Okay. Jessica, did you have me for the first one? Michelle, did you have me for the first one? Okay, Heidi. All right, cool. Okay, cool. All right. Okay. All right. Catherine. Okay, cool. All right. So let's see. You probably had Dr. McCain. She's the other one for 137, Michelle. Okay. So we have the quiz that opened up this week, right? If we haven't taken the quiz, okay, cool. If we haven't taken the quiz before this ends, please, sometimes I remember, sometimes I don't, remind me to send out the attendance sheet for everybody, okay? Remind me to send out the attendance sheet. It's a little link on here, and that's how you guys get credit for attending this spotlight session. Okay, so the quiz is opened up this week. It's covering Lab 1. It's a 20-point quiz. If you haven't taken it already, there's 20 questions, about 3 conversion questions. There are 3 or 4 baby questions, I think like the neck, shoulders, and cheek area. I looked over there's some quadrants on there that I harped on through my labs. If any of you guys have me for lab. Does anybody have me for lab? I think some of you all do. Okay. Okay, so you've, yes. All right, so some of my people, okay, some of my people probably heard me harp on it. Okay. Karen Drake, yeah, I share a Blackboard shell with her. So, okay, cool. Okay, well, so like I said, 3 baby questions, 3 conversions, quadrants. Know your quadrants. My folks know how I feel about quadrants. They're important. Make sure you know the regions and the quadrants. I know there was a student that asked both, and it's both. And then there's going to be questions like parts, like what if they point to a kidney or they point to a stomach, it's going to ask you what part of the system is it in, okay? Or they're going to point to an organ, and you're going to have to identify that organ. All right, all of this can be found in Lab 1. Does anybody know where the hair, skin, and nails, what system they're part of? That's right, Brooklyn. Yes, that's right. I don't know if some of you guys had me in class tonight, but we already kind of hammered on this. That's right. Okay, everybody's coming in. Awesome. Very active tonight. That's what I love to see. Okay, so yes. So another question. Let's talk about test taking skills real quick. This is a timed quiz. Okay, it is 20 points. You do not want to spend time. You don't want to spend eating up time on questions that you don't know about. Okay, you don't want to leave 3 or 4 unanswered. That is bad news. That'll drop your grade down a letter, if not more. If you do not know the question, move on. Do not sit there or second guess yourself and eat time up trying to figure it out because you're flustered. You know, just calm down and try to get a quiet space. Let's see. You know what I do when I take stuff? Hold on, I'll show you. So when I was a student, I put these on, and they're my gum muffs. I would sit in a quiet room, and this would help me concentrate. So I recommend finding something. I know we're working from home or working from the office or even from the lab, but I encourage you guys to try to find a quiet space and get focused when you take these quizzes, because they're timed, and sometimes people need to learn how to take timed exams. Chances are when you reach your licensure exam way down the road, it's going to be a timed test. And what better way to start developing your test taking skills under a clock than to on Bio 137. So my next point, if you have an answer choice that doesn't make sense or you've never heard of it, but it's like too easy to be true, chances are that's the wrong answer. So if we have a question that says, in what system is the hair, skin, and nails components in, and you have all these different choices, and you have one, I guess I'm talking about hair, let's say hair. Let's say what part of under what system does the hair belong to? And one of the answer choices is follicular. Well, if you think, okay, follicle, hair follicle, follicular, that makes sense. I'm going to click that and go on. You would be answering incorrectly. Okay. If it's for the people who don't know, who guess, and because they think it sounds right. Sometimes that works. Oftentimes it does not. Follicular is more pertaining to reproduction, follicle cell reproduction cells, particularly in females. But that's pretty much why we use follicular. So don't, just because it may be like the easy one that we don't know doesn't mean it's going to be the correct one. So I don't want you all to get baited on that. I think I've covered the quiz. The deadline is on Thursday. So, and I have I gave my students the lab key, the lab 1 key. I'm not sure if you guys have received it from your lab instructors, but if you're interested, just send me an email and I'll be happy to send that out to you. This is 201 PCTCS. All right, that's me. So let me know. I think at this point we have a good understanding of the syllabus, especially for some of you people who have probably taken, this is your second one. So let me know if we have any questions about syllabus. What I really want to probably talk about is lab 2. All right. Lab 2 basically is microscopy, and I had my in-person lab tonight, and everybody seemed to know. Everybody seemed to have an idea what was going on, so that's good. I think I'll make sure I don't miss any chats here. Okay. So on the lab 2, what you probably want to focus on is understanding the. Let me see if I can mirror my screen here. Okay, I think you guys can see this. So we want to understand the microscope, and this is memorization. Okay, so down here are the parts and functions, and I want to draw your attention to the condenser, okay, and the iris, the objective lenses, and the ocular lenses. Those are ones that I've seen show up on quizzes in the past, and in all likelihood they may show up on this year's quiz also. The condenser is under the stage. The stage is what holds the slide, but under the stage is called the condenser, and the condenser helps modulate or control the amount of light coming through the stage to enter your slide, your target sample. And sometimes when we have too much light going through, you can pretty much bleach out everything that you see. All right. For example, like epithelial cells are really transparent. So we want to really tone down and provide contrast to the cells, and that way they are able to you're able to see them. If you bleach everything out, you don't see anything, and you could possibly be missing what you're looking for. So the purpose of the condenser is to control the amount of light coming through, so you can pick up on cells that may be hard to detect. So keep track on that. Now at the top are these ocular lenses, right? They're up here. And these are under a magnification. All right. So if a question were to ask you, what is the total magnification that you're using? You cannot forget about this right here. Okay. This is your ocular lens. That is a 10x magnification. Okay. So already you're already looking at 10 times the magnification while using this ocular. Now, objectives. There's about 3 or 4 that you guys are going to see. If you're in person, you're definitely going to see it. But if you're online, then this, unfortunately, this is the closest we can get. Objectives are different magnification tools at different powers. Okay. There's a red one that we can't really see. That's in the back. That's going to be 4x. That's considered low power. The yellow one is considered still low power. It's a 10x. So 10 times. No, excuse me. Yeah, I guess no. It's 40. No, this is 40. It's 10. Excuse me. The yellow one is 10x. All right. So 10x more magnification. The blue one here is 40x. That's what we consider high power right now. High power is 40x. Okay. So if we go back to the question we asked, what is the total magnification? You're going to be, well, Drew said it was the ocular, which is 10x. And then depending on what objective we're using, you multiply that 10x by either the objective, which could be 4x, 10x, or 40. Okay, so if we're under the 10x objective, all right, listen up. If we're under the 10x objective, what is our total magnification? Somebody chat me something, or speak out loud if you'd like. Yeah, that's right. It's 100. And if you want to really get it right, we say, excuse me, I put 110. It's 100x. Put the x in it. That means magnification. So, but yes, I'm Maytap, perhaps Maytap. I think that is that's the correct answer. Did everybody else see that? We got 17 people in here. I have one person respond. Is everybody just blowing me off? Does everybody understand that? Let's do another one. I want to get some more responses. Okay. Now, let's say we're under the 4x. Now, I want some people to respond back. What's the, yeah. All right. All right. And then let's do 40x. What's the 40x? Yeah. Okay. Yeah. Put the x behind it. Some people will knock you on that. I won't, but you'll that is nickel and dime credit right there. You'll get half credit for not putting an X. So please remember the X. Some people are sticklers about that. Okay. That is part of the lab we're going to be doing on, that's a big one. Okay. Now, there's some other things that we want to talk about. I want to talk about structure, cell structure. And I'm going to get down to that. And that is, okay. I want to focus on page 213. Page 213, structure with cells. If you were to look at certain cells in your body, you will understand that they're all different shapes and sizes, okay? And the thing that you want to try to remember that tends to be a theme throughout your clinical careers is that cell structure will yield its function. Now, what do I mean by that? I mean that judging how the cell looks, the shape of it, the size of it, will kind of tell you what its purpose is, what its function is. So structure yields function. If you were to Google image search what a simple squamous epithelial cell looks like, you're going to find a cell that is more like a square. Okay, square, rounded, maybe it's kind of blocky shaped. And what simple squamous cells do is really they're like a barrier, like an extra barrier. They'll find them in mucosal linings. They're just more like a kind of like your skin layers, epithelial cells. They're like they help as a barrier. There's really no else to put it. Cardiac muscle cells. Cardiac muscle cells are like really long. Let me see if I can Google something. Let me see, and I'll share my screen. Yeah. Okay. So if I were to just do a Google image search, and I encourage you guys to do this. They look kind of long and narrow, and they pinch off closer at the ends. Like this is a good example. This is a good example. Here's another one. They run together, and if you understand, let me go back to you guys, and if you understand what cardiac muscle cells do, what's the heart do? Tell me what the heart does, or we're going to have a long night. Pumps. Yeah, it pumps. Okay, it pumps. So how does it pump? Circulates fluids. You're right. How does it pump? It pumps. Let's say what happens when your heart stops? You go to the ER, they put the paddles on you and they shock you, right? Yeah, it contracts. How does it contract? You may not know this. It contracts through an electrical conduction. Okay, when your heart stops in the ER, hopefully they can put paddles on you and they shock your heart back into rhythm. Okay, you go into what's called an asystole, which means there is no systole, there is no contraction. If they shock you, they administer an electrical conduction through your heart that will help contract these cardiac muscles so you can pump blood through your body. Now, luckily, our hearts have their own electrical conduction device called the SA node, located in your left upper atrium. Okay. Now, when that happens, it sends an electrical conduction through the cardiac muscle tissue that helps contract the cardiac cells, which causes your blood to move out. If you were to remember those images that I showed you of cardiac muscle cells, they are designed to propagate this electrical conduction through all the cells. Okay, they connect to their friends, and it pretty much offers a low resistance pathway of electrical conduction all the way down to the bottom of the heart. Because if you look at the heart, all the valves are at the top. There's no valves at the bottom. So what we want to do, it's like we want to squeeze the bottom of the heart so we can inject the blood out. So electrical conduction starts at the top, travels all the way down, activates the cardiac muscle cells to contract, and then blood is ejected out into the aorta or pulmonary vein. Okay, to the lungs to get oxygenated if it's deoxygenated. So the purpose I'm trying to make here is if you look at the shape of these cells, will kind of dictate what their function is. They're long and narrow. They're used to propagate an action potential all the way, the same thing as electrical conduction, to the heart. All right, human red blood cells. Does anybody know what a red blood cell looks like? Tell me what it is. Either speak up or put it. Yeah, it's a donut. Brittany, did I have you tonight? Did I have you guys tonight? All right. Yeah. Okay. Yeah. LOL. It's like redundancy. Or Groundhog Day. All right. So it's a donut, right? It's a donut, and in the middle. I'm glad you all listened. At least you can recall that. It makes me happy. It's not like I'm just up here talking to a wall. So yeah, you remembered it. It's like a donut, and what happens is that in the middle of this donut, that's where we carry the oxygen, right? So if you all ever want to Google image search what a red blood cell looks like, it will be kind of hollow in the middle, like a donut, and that's where your oxygen molecule goes to go to tissues or go to the lungs to get more oxygen. All right. Now, our last one, sperm cells. Sperm cells, we have an idea what sperm cells look like. They have this long flagella-like tail on the back of them. Now, for some of you all who had me tonight, you can't answer these. What is, unless nobody answers, and you can at least help me out and don't leave me hanging. What is the organelle of the cell that we call the powerhouse of the cell? Think about that. What? Yeah, that's right. The mitochondria. Okay. Yeah, good job. Thank you. The mitochondria. The interesting thing about sperm is sperm are used to go fertilize eggs, right? They have this tail, and they have to not only fight gravity because they have to swim up the vaginal canal. So not only do they have to fight gravity, but they also have to move against vaginal diffusion, which is sometimes low, going from high to low. So they have to fight through gravity, and they have to fight through, just the normal flow of the vaginal canal, which is to move out. And how they do this is that they have a lot of mitochondria to make enough energy to propel the tail, the flagella, up the vaginal canal into the fallopian tube to fertilize an egg and create a zygote. So if you think about it, that's why sperm cells have a lot of mitochondria. They're loaded down with mitochondria because they need the energy to swim up into the fallopian tube to find an egg. So it's kind of interesting. The point I'm trying to make here is that structure yields function. All right, if you look at how the structure of the cell looks, you can kind of dictate what the purpose of that cell is. There's a lot of other different examples, but those are 4 of them that I kind of wanted to cover. All right, so that is at the very end. That's on page 213. I want you all to check out the cell structure on page 212. Make sure you know the difference between rough endoplasmic reticulum and smooth endoplasmic reticulum. A lot of this may be some high school biology that we might have had last year, or if you were like me, you went out into the field and you spent some time between classes, or at least from high school. Okay, now I want to talk about a few things on the lab. Now, this is not just lab. If you all have questions about lecture, or if you have any kind of questions about what we're doing in the lecture, I'm here to help with that. I kind of encourage you guys to speak up, because this is what these are about. Or if you have a question and you want to chat it, chat it, and I will be happy to help you out. But if we don't, then I'm going to kind of give you a preview of what you're going to be talking about in the lab. Okay. So when we talk about 4x, 10x, and 40x, we need to understand what we're talking about. We need low power or high power. All right, low power is going to be 4x or 10x, and then high power in this case is going to be considered 40x. So you may hear that term. Now, tonight we talked about, let me see what I can find. I keep lost my track. The lens effect. The lens effect is basically, it's like a physics concept where I cannot remember how it was. I got it confused in class tonight. It looks like an upside down E when you're looking at, like, we have a slide that has a letter E on it, a lowercase e. So we all know what that looks like. When you look at it with your naked eye, the E is upside down. When you put that E underneath the microscope, it is flipped, and it looks like it should, like a upright E. Not a backwards E, if that makes sense. A right side up lowercase e. And that is called the lens effect. And that's just how microscopes work. It is the mirror image of what you look like, what you see with your naked eye. Another thing that I want to touch on is called the depth of field. And when you look at a microscope and you get your hands on these microscopes, you realize that they focus, and everything is 3D. Okay, everything is 3D. You have to understand that when we're looking at cells, everything is a three-dimensional plane. So when we focus, we are focusing through these planes. So some things may be focused and some layers may not be focused. It's like layers of cells. And the reason why we do this is because, especially in histology, when I kind of talked about this last week in some of my classes, where if we were to take a tumor or a piece of tissue out of a person and we send it to the pathologist to see if it's malignant or how bad the invasion is of malignant cells, they would look at the margins, these cell layers. And when you think of margins, think of it as like looking at your fingerprint. You know how everything's kind of uniform there, and they all kind of nicely smooth together, the ridges of your fingerprint. This is kind of like how cell tissues are. Well, when there is a malignancy, it doesn't have these margins. We're going to call them margins, and they kind of branch out. And what happens is that that's how you can kind of tell if you've gotten all the malignancy or not when you do like a biopsy or you pull it all out. Pull it all out, I mean pull all the tissue out. You can look at these margins and see, okay, yes, we have bad margins in the middle of some good margins, so that tells us that we got the whole tumor. And if you've taken more out, then, or you've taken some out and you still have these bad margins, these malignancies, this abnormality margins, then that says that you need to take more tissue out. Unfortunately, you need to go deeper or take a lot larger amount out. So that's kind of another way we use microscopes. I told some folks tonight if we wanted to look at viruses, we would need a much more higher powered microscope, probably like a electron microscope that cost nearly, could cost $500,000 up to $1 million. And there's different versions of microscopes out there. Ours is just a simple light microscope, still worth a couple $100, I think. I mean, they're good scopes. Okay, so I'm trying to think what else I want to touch on. If things are highlighted or bolded in your lab manual, they will likely make their way on the quiz or exam. So we want to touch on that. I just talked to you about the depth of field, which is the different planes. So be aware of that. And you're going to do an exercise on that. And then at the very end, and we already talked about scanning low power and high power. That is the table on page 210. And you'll probably talk more about that. If you have any questions, check in with me or your lab instructor, and we'll kind of take it from there. And then you're going to do what's called a wet mount at the very end, which is kind of like where you rub your cheek on a slide and you try to get it focused. So some of you guys are taking this online. Eventually, you will have a chance to work with a microscope, but I do encourage you at some point to get familiar with it, because when you reach the higher levels in micro and stuff, yeah, and the lab manual, Michelle, I'm looking at the lab manual, page 211 or 210. It's like 2-10 or something like that. And when you get to micro, you're gonna need to know your way around. No problem. You know your way around a microscope. That is all I think I have prepared tonight. A lot of this is I mean, I could rattle on, but what you really need to do is kind of get in the, be able to, for online, obviously learn what I talked about earlier, learn the objectives, learn how to do total magnification. If you were given a question, or be able to describe the depth of field, or the lens effect, which is the upside down E. Okay. And I'm trying to think something else. I don't think I have anything else. And the organelles of the cell. I told you the tips for quiz 1, right? Are you asking about quiz 2, Michelle? I don't know if you came in late. Okay, quiz 2. Okay, so you heard me. You heard what I said about quiz 1. So quiz 2 will be a 10-point quiz. Quiz 2 will be a 10-point quiz. It'll be the different components of the microscope. There may be a question on total magnification. And I think after I kind of poked everybody, they gave me some answers, 40x, 400x. There's going to be that question right there. You need to remember that the oculars have a 10x magnification. It's going to be multiple choice. It's going to be timed. And then there may be a couple questions on what organelle this is in the cell. Last year it was centrioles. Like they'll point out like an arrow on the picture of a cell that says what organelle is this, and I think it was centrioles. But make sure you're able to identify the different organelles of the cell. Golgi apparatus, mitochondria, the difference between smooth and rough endoplasmic reticulum. And I think that'll be the majority of the test, of the quiz. Yeah, we can talk about chemical bonds. That is like, we could be all night on chemical bonds. I'll talk lecture. Is this chemistry, atoms, atomic theory, and all that? Chapter 2? The whole thing. So protons, neutrons, electrons. Okay. Well, y'all should have spoke up earlier. We could have gone right into that. We didn't have to talk about boring microscope. Protons. Now you have to help me what you want to know, because you're giving me a large topic to talk about. So I can give you my spill, but if you want something pointed, you have to ask me, and I have to get out of here at 9:45 because I got to go to work at 10. So now we can always continue this later on down. I got another one at 8 a.m. on Wednesday. We can always continue this conversation if you guys want to check in, but this is what these spotlights are supposed to do. So let me get to it. Ionic bonds. Covalent bonds share electrons. All right. So if you were to have covalent bonds are going to be like what we call polar atoms. All right, polar elements that are going to like share the electrons between two atoms, which create the bond. Bonds are pretty much made out of electrons. Ionic bonds grab all the electrons. Okay, there are some elements on the periodic table are more willing to give up electrons than they are to take electrons. All right, like for instance, NaCl, table salt, right? Na is sodium. That is on the far left-hand side of the periodic table. Elements over there are more willing to fork up an electron to somebody else because that's just the way they're built. Chloride is on the far right side of the periodic table, and they want to satisfy what's called an octet, which is all the electrons around them. They're greedy. Chloride's greedy. So it wants all the electrons. So it will happily take on another electron to satisfy its octet rule, which is 8 electrons. The way chloride is in the periodic table, the column that it's in have 7 electrons. Okay? What else is in that column, guys? Fluoride, chloride, bromine. What are the halogens? Is everybody following me? You have the noble gases on the far right, neon and all them. They don't do anything. They're called inert gases. The column next to it is called the halogens. That is fluoride, chloride, bromine, iodine, maybe a few other ones. They want an extra electron to satisfy their octet rule, otherwise known electronegative. Have we heard the term electronegative? They are electronegative elements. They will gladly grab an electron from sodium or anything on that left-hand side. The sodium's one, magnesium, calcium, potassium, lithium, beryllium. Are you following me? And that is what creates, okay, all right. I have to know because I don't, all right. I don't see any heads moving. If I hear silence, it scares me. Normally I can see through expressions and I can help. So with ionic bonds, that is pretty much one element stealing electrons from the other one. There's no sharing going on. That is chloride being greedy and taking all the electrons from sodium. But sodium's happy to get rid of them. Sodium wants to be a +1 charge. Does that help answer your question? As in, chloride likes to be negative because it likes to take on an extra electron to satisfy its octet rule. So do we understand that? Okay. Okay, Brittany, we're going to you next. This is recorded, guys. So I'm going to throw this up. I have a YouTube account now because some students wanted some accessibility, and this was my answer, was YouTube. So now I'm on YouTube. So it's kind of different watching yourself on that. So I can post this on YouTube, and if you want to go through this again, this is how I learned. I watched and recorded all my lectures, and I went back and wrote it all out. That's how I got through school. So if you want to check out the YouTube account, go for it. Find this spotlight session. I'll probably have it up before 11 p.m. tonight. I got to go to work and touch base with some people, but then I'll have it up probably between 11 and 11:30ish. So FYI. Brittany, oxidative phosphorylation is something I'm confused about. Okay, well, I'm going to call it OP. OP is involved, but the interesting thing about oxidative phosphorylation is happens in the mitochondria, right? And this is kind of what we talked about with the sperm. Oxidative phosphorylation is the last steps to making what we call ATP. ATP helps is pretty much the energy money of the cell. You know how we have money to buy drink and food and get through life, dollar bills? We have to go out and earn. The cell has ATP, and it has to use and give ATP to do things that it wants to do. All right, adenosine triphosphate. Okay, adenosine triphosphate, otherwise known as ATP. Oxidative phosphorylation is the last step in the mitochondria that takes these electrons that we talked about earlier, and these electrons do these different things, and it will create ATP. Okay. Now, you know how, let's say you have $5 and you buy a drink and you get some change back. Well, let's talk about it like this way. If an ATP, if you're a cell and you need something, you give mitochondria ATP and you have some change left over. All right, that change is called ADP, adenosine diphosphate. So we talked about 3 phosphates, now we're down to 2 phosphates. All right, ADP is pretty much what's spent. That is like you getting your change back after you got your drink. You got some change back. Well, the cell gets adenosine diphosphate back, which is pretty much useless. It doesn't have a lot of energy to it. It doesn't have a lot of value to it. What oxidative phosphorylation does is that it will tack on that 3rd phosphate back on to make it ATP. Okay? Does that make sense? Do we need to go more in depth than that, or am I off on that? I'm trying to figure out, I got to figure out how much we need to know about oxidative phosphorylation. Okay, it makes ATP. It goes through different complexes. I mean, we can get into some biochemistry, but I don't want to blow everybody's tops off. Do we need anything about complexes? Okay, does that answer the question? Okay. Because if we need to go more in depth, we can. But like I said, I want to stay within the scope of the class. Okay, so I guess we're okay. Is there any other questions? if you want to understand where OP, oxidative phosphorylation, comes in on, you have glycolysis, which is first. You have what's called the citric acid cycle. And then from the citric acid cycle is when you start oxidative phosphorylation. So all these different steps come down before we can get any ATP out of a cell. Everything happens in the mitochondria except glycolysis. Glycolysis takes place out in the cytoplasm. Chapter 2 is probably one of the tougher chapters. I remember tutoring this class a long time ago, and you know, chapter 2 they hit pretty hard with biochemistry and just general chemistry. So, is there any other questions about chapter 2 or any other topic you want to talk about? We have 15 more minutes. We can get a lot in. Okay. Okie dokie. All right. Well, if something comes up, let me know. I'm going to go get this attendance sheet, and I think that's all we need to talk about today. Let me find it on my email here. All right. I heard those chats. Hold on. I'm going to come back. Yes, I got the sign-up sheet coming. Yeah, Melissa, but normally I have my I think you have, I don't know what the channel is. I mean, I'm like a novice at this. Just Google my name. Google like Bio 137. You should see it. Google that. Here is the attendance sheet. Work through that. I'm going to hang out and answer some questions. Michelle, I wish I had a better answer for you. I don't know. I just Google it and I find myself on there, and you can like go into my channel. Some people have subscribed. Some of it's cool. Some of it you probably won't care about. How many minutes are the exam quizzes? This one's going to be 10 minutes or 12 minutes. I can't remember. It has Meredith. It has the directions on there. How long the quizzes are. It'll say this is a 10-minute quiz. Please read all that before you start it, and it'll give you like a breakdown. Oh, and Melissa, yeah, I can make YouTube videos of pretty much anything you want through these spotlight sessions. If you have something of interest, this is what these are for. These are for you guys to pretty much what we called it in graduate school is getting pimped, where you sit here and you just hammer me with questions, and I'm here to respond. So if you want to, it's called getting pimped, is what it's like, an old school term that a lot of people don't think of it in that term, but it's you sitting here and peppering me with questions. And this is what these are for. So if you want something that you want to talk about, you're confused about, please let me know and we can discuss it, and hopefully things will look more, you'll understand it more. Okay. Yeah, 10 minutes. All right, cool. And so let me know when you guys are done, and especially you first timers that are doing the attendance. I'll do it like, okay, thank you, Alyssa. You all are going to make me a star. Put it as in like Drew Mattmiller Monday night. Like, I don't think the title's a big deal. Put Monday night with Mr. Mattmiller or something like that. And you can put the date on there, 1/25/21. Oh, and John, if you got it all finished, then you guys are good to go. Once you do the attendance form, you're good to go. But I want to hang out and make sure everybody can do it, especially our first timers. And you can sign up as many times as you want. Okay, thank you, Kayla. Cool. All right, Brendy, sounds good. See you guys next week in Monday Lab. Thank you. Put me, because I'm your spotlight. If it asks for course instructor, then I guess put your lecture person, or you can put both. Give them as much information as possible, so they know what they're doing. Caitlin, that was to answer your question. Okay, thanks, Tony. Oh, Bethel, the course section is going to be found on your Blackboard. Like when you click on your class, it's going to be like a 4-letter, like 19Z3. Check on your Blackboard real quick. Bethel, that was to answer your question. Okay, cool. I don't know any details about the lecture exam, Jessica. I don't. I think it'll probably be. You can ask your lecture instructor. It may be detailed in your lecture syllabus how the exams are. And Kayla, I think they do. Kayla. I'm here to answer both lab and lecture. I've tried to get some clarification on that, and I haven't yet to get an email response. So I'm going with the fact that these count for both lecture and lab. Caitlin, as long as you submitted it, it's fine. I think you're going to be okay. Just, if you feel like you need to keep coming back, please come back. And then you guys are free to go once you get that attendance form filled out. But I'm going to be here until everybody gets it. I can see her looking fast in her faded jeans. Thanks, Brenda. See you later. All right, Jessica. Have a good week. Stay dry. Okay, Michelle, I'll check my email. I got to go to work at 10, but I'll check it. I'll get you what you need. Come check in later. Thank you, Meredith. You too. Okay, good, Bethel. It sounds like you got it right. Or you found the answer. Alyssa, are you there? Oh, okay. I think it will. Yeah, I think it will. If it took it, then you're fine. They'll find you. And if not, you may want to submit an email to your lecturer professor. But I have you on record here. If you need me to vouch for you, I will. But just let me know. If it took it, then I'm assuming it went. Okay. All right. Well, I'm gonna end it. Enjoy the rest of your week, and we'll see you next time. Peace.